Thank you all for joining us today for our May session of the Torrey Talks lecture series. My name is Jordan Hoffman. I'm the programming chair for the Torrey Botanical Society, and I'm happy to have you all here. So today we're going to continue this ongoing line of exciting talks that we've had for this year with our current guest speaker, Dr. Robin Sleith. So after getting his bachelor's in biology at Green Mountain College, Robin went on to do his doctorate at the City University of New York, where he was working with Dr. Ken Carroll at the New York Botanical Garden. After graduating in 2018, Robin went on to continue his research through postdoctoral positions. So he's currently a postdoctoral researcher at the Bigelow Laboratory for Ocean Sciences, but he was also at Smith College before this. So Robin has explored a lot of really interesting questions and topics involving algae, which is a particularly understudied group of cryptogams. And throughout his research, and today he's going to talk to us about some of his current work on the symbiosis between green algae and testate amoebas. So with that, I'd like to turn it over to Robin.
Awesome. Thanks, Jordan. Thanks, everybody, for attending. So it's great to have the opportunity to share some of the projects I've been working on since leaving NYBG. And so I'm going to start by talking about some work that I did at Smith College, looking at this testate amoeba that has these microalgae, Chlorella microalgae, and trying to understand just the relationship between and within that system. And so two people I really want to highlight from that work are Billy Cullison, who was an undergrad student at the time at Smith College. Don't know where she was getting to hold this koala, but pretty amazing and maybe not safe. But then Agnes Viner, who was another postdoc at Smith at the same time, who really helped to spearhead this effort. And then we'll sort of transition to talk about some of the work that I've been doing here at Bigelow using eDNA tools to look at an algae bloom in a freshwater coastal lake. And so that was work that Sharon Mann here has been doing. She's an eDNA grad student at the University of Southern Maine. So to get started, I'll just sort of introduce this system. So this is testate amoeba, which is in the amoebozoa in this Arcellinida group. And this particular guy is Hyalosphenia papilio. And what's really great about this guy is every time that you find it in the wild, it has these green symbionts in there. And I'm putting symbionts in quotes because the jury's still out on what we really want to call these things. And so this right here is the organic test or shell. So they're called testate, which basically just means shell. So they build this shell around themselves. And then the actual amoeba, you can see, it's inside here, but then it has these pseudopods that are sticking out. So this is the cytoplasm of the amoeba in here. And in that cytoplasm, you see these green algae. And so we were interested in understanding, first of all, what is the diversity of these testate amoeba in the Northeast, and what's the diversity of the Chlorella in those amoeba? And then investigating this relationship between the host and the algae. So trying to figure out, is this a true symbiosis? Is it something more like kleptoplasty, which is when a heterotrophic organism basically will just steal the chloroplast and not the entire, it might just take that chloroplast, ingest the rest of the cell and keep that chloroplast and gain some energy from it for a brief period of time, and then just sort of it has to keep ingesting those. So we don't necessarily think that it's a kleptoplastic relationship, but that's just sort of an illustration of this sort of spectrum. And that's what this figure on the right is showing, from heterotrophy to phototrophy, different organisms. Most of these are microbial eukaryotic organisms, and how they are associated with phototrophic partners. So you can have things that are rarely associated to things that are always associated. And then it gets into this really interesting, deep time evolutionary piece where if this relationship lasts long enough, maybe you become a permanent endosymbiont. And if you're a permanent endosymbiont, maybe eventually you're becoming an organelle, a plastid at that point. And there's that example of Pollinella where we think we're seeing sort of the formation of a plastid happening at this time point, rather than there are only so many examples that we can find throughout evolutionary history where that has happened. And then finally, we just were interested in what happens to these chlorella cells over time when the amoeba are stressed out. And so just to sort of illustrate some of our expectations or questions going into this. So here we have the amoeba, and then in the cytoplasm, you have these chlorella cells, and then the chlorella nucleus is orange or brown here. And so if you expect the chlorella to be sort of active and living their best life inside this amoeba, you would expect if you basically ground up this whole thing and looked at the transcriptome, you would expect there to be a mix of amoeba transcripts and chlorella transcripts, right? Because these algae are, undergoing all of their norm. If it's an entire intact, viable, living algal cell, it should be undergoing normal processes. Yes, it might not have to deal with some of the, factors that it would have to deal with if it's free living, but we would expect maybe photosynthetic genes to still show up. Versus if something else is going on, if it's more towards the kleptoplastic side of things or some other relationship, maybe that chlorella nucleus is not active, and so we would just see amoeba transcripts and very little evidence of algal transcripts. Okay, so how do we go about asking those sorts of questions? First, we have to go out and get these amoeba from the environment. So this is the really hard part because we have to go to beautiful bogs, and you know me, I just can't stand tromping around a bog and looking at all of the amazing sphagnum and other things that are growing there. So we had two nearby bogs, one in right near Smith College in Western Mass, and then one a little further east at Harvard Forest, and then two in Maine. So one up in Acadia and one in Orono. And so several of these are public accessible bogs. There's a great boardwalk in the Orono bog and in Holly bog. So I would highly recommend, if you find yourself in those areas, look up those bogs and go visit them because they are really great places. So then we collect, basically the way we collect these is by collecting sphagnum moss. So we'll just put some sphagnum moss in a Tupperware, bring that back to the lab, collect some water from that bog as well, use that bog water to rinse off the sphagnum and basically sort of shake that around, and then all of the amoeba will fall off of that moss and into our petri dishes, where we're able to keep them alive for a number of days, but not much longer than that. Maybe weeks at some time, but you certainly can't get a really stable culture of these guys yet. So then when they're in those dishes, we're able to either do our experiments or harvest them for single cell whole transcript amplifications, or WTAs, or single cell whole genome amplifications, or WGAs. And so that involves someone who is much better at picking cells than I am, who will go in and just grab one of those amoeba and put it in a tube, and then you're just doing basically the reaction in that tube to lyse and amplify, and then we make a sequencing library from that and send that out for next generation sequencing. And that's a method that Laura Katz's lab at Smith College has been doing for quite some time, and has a lot of experience with both of those methods. And the way that they analyze that data, because you're basically getting these really diverse communities. Whenever you're collecting these microbial eukaryotic cells, whether it's a ciliate or whether it's one of these amoeba, first of all, there's going to be lots of bacterial contamination on there. And second of all, which you can sort of take care of with the WTAs if you're doing a poly-A selection. But second of all, there may be other eukaryotic, like microalgae in this case, or just other organisms in there. So it really takes a, you know, a really fine-toothed comb and curated approach to sort all of that out. And so the way that they do that is a phylogenomic pipeline that has been developed in that lab that I'd be happy to talk to folks about, because it's a pretty useful, I would say, and published pipeline that can, is a pretty helpful way to make sense of these really complex data sets. And then, so we have that method, and then we're able to do a little bit of read mapping on a more sort of as needed basis to zero in on some of the patterns that we're seeing. And then we were really lucky to partner with some folks at JGI who are experts in TEM and a lot of these staining techniques to actually start doing some imaging of the amoeba cells to see what the algal cells in that amoeba cytoplasm look like, and whether there's nuclear material, all of those sorts of things. Okay, so I'm going to jump right into some results. So here we are surveying the Chlorella diversity. And so we do this by collecting the RBCL region of the chloroplast genome from WGAs of the testate amoeba. So we're targeting the Chlorella and the testate amoeba, but what we do is we pick the testate amoeba, do a WGA of that, and the Chlorella in that testate amoeba comes out of that WGA. So then we search for the RBCL signal in that mess of everything, pull that out for the Chlorella, and build a phylogeny of that. And so what's really interesting is that I'll walk through this figure real quick. We have host organism here. So this testate amoeba represents all testate amoeba, whether it's Hyalosphenia or Arcella or some of the other lineages in that group that do harbor green Chlorella symbionts. And then if it's this is Rhizaria, which is another group of microbial eukaryotes that have Chlorella symbionts. And this represents ciliates, which mostly is Paramecium bursaria, which has these. And then this is going to be if it's free living. Then we have our study sites, so Harvard Forest, Holly Bog, Orono Bog, and Acadia. And then the distribution from the literature or from this study. And so this testate amoeba symbiont clade 1, TACS1, is what was described in the literature. And they found it in testate amoeba and in Rhizaria. And they find it in Asia and Europe and in North America. We do detect that clade in our data set, but only at Harvard Forest. And then we also detect this testate amoeba symbiont clade 2, we're calling it because we're very creative. And we find that just in testate amoeba because that's the only thing we were looking in. And we found that one at Holly Bog, Orono Bog, and Acadia, and those are just in North America. And so what's interesting about this is, number one, there was previously a paper called One Alga to Rule Them All that had done this sort of global sampling looking at this Chlorella across different organisms to see what kind of diversity existed in that Chlorella. And they were really amazed that across the amoeba that they looked at, whether they were Rhizaria or Arcellinida, they only found this one clade. So that's why they were saying one alga to rule them all. No matter what kind of amoeba this is in, we're finding this one clade of algae. But we go out and we survey the eastern US. They didn't, I think they mostly were surveying western US, and we find this other clade. So that's just interesting and increasing the amount of diversity of Chlorella. The other thing is that it's not monophyletic. So this does appear to be sort of an independent acquisition from the environment. So that's cool. That's step one. So next we looked at the amoeba diversity. So now we're looking at COX-1, a mitochondrial marker, and we see that we have some geographic clades here. So the clade with the alphabet is from the existing literature of what's known of diversity in this amoeba group. And so you can see like these are all GenBank numbers, and then anything that's LKH is from our study. So you see that clade M here is only found in Harvard Forest, and clade K here only in Acadia, and then you have clade F at Holly Bog and Orono Bog. And so first of all, you can see that this TACS2, you could say like, oh, well, maybe this pattern is that the amoeba diversity is tied to the Chlorella diversity, right? So there's like coevolution occurring, which you would expect to happen if these were vertically inherited symbionts, right? If it's getting passed down from generation to generation, then I think you would expect the relationship of the Chlorella to mirror the relationship of the amoeba. But we don't see that here because you're seeing these TACS2 in clade F and clade K. So then our friends at JGI who do amazing work took a look at this under TEM. And so first here we have autofluorescence. So this is just gonna show you where those Chlorella cells are, because those chloroplasts are going to autofluoresce. Then we have DAPI staining, which you can see also is doing a good job lighting up the Chlorella cells. And then you can also see the nucleus of the amoeba is here and really cool and crazy looking, and some folks are working on that. And then if you subtract that autofluorescence out, you can see the Chlorella nuclei lighting up here, and then the amoeba nucleus here. And then you put it all together and you can get this sort of idea of where the algae are, where the nuclei of the algae are. And then we can zoom way in. And this is the actual TEM here. This is just confocal, I believe. And so over here, you're able to see that this is sort of like a pretty intact Chlorella cell, fairly similar to what you would see in a free-living Chlorella. So nothing super strange standing out from the microscopy. So then trying to look at the Chlorella relationship with the host through this transcriptome. And so we're trying to identify Chlorella transcripts from those WTAs, and we just do not find very many of them. We're doing a really good job finding amoeba transcripts. And so then this is an example where a reviewer was pretty helpful. They were saying like, okay, well, imagine if this is some incorporation for photosynthesis, like sure, we would maybe not expect to see a bunch of the Chlorella housekeeping transcripts because maybe its function is so different when it's incorporated, but you would still expect to see photosynthesis transcripts, I would imagine. And so we just don't see a lot of evidence of that. So that leaves us with one more piece to look at, which is that this experiment where we basically starved the amoeba by diluting out the media and photographing them and then taking a sample for whole genome amplification every few days. And so day 0 here, the amoeba are looking healthy. The Chlorella in the amoeba are looking healthy. This is what we're used to seeing when we're looking at these guys under the microscope. And we also see this is coverage of the chloroplast genome. So it's basically saying, like, at any given place on the chloroplast genome, you have 60x coverage. That's covered by 60 times, which just means there was a lot of chloroplast signal in that WGA. And then as you go out in time, almost immediately that chloroplast signal drops off, and you can start to see by day 12 that those algal cells are really getting quite degraded. So this is, it's fairly, it's quite expected, I'll say. If you have all of these basically powerhouses inside of you, it's great when everything is going well for you to use those as, photosynthetic energy generating resources. But if you're being starved and things are not going well, the first thing you're going to do is you're going to eat those up and digest them for all of the valuable nutrients that are in there, rather than trying to maintain these things. So just a sort of interesting additional test that we did on this system. So conclusions for this, we did find that there are these diverse Chlorella strains, and we basically, I'm not sure that we really clear up what kind of relationship this is, just to say that it's not as mutualistic or mutually beneficial. If it seems like the Chlorella, something's going on, they don't seem to be, happy little cells doing their normal intracellular, song and dance. So really what we want to do to move to the next step with this is to get a stable culture of some of these amoeba. So you can imagine how valuable it would be to do sort of like cross-infection experiments, where you could take one of the TACS2 Chlorella and try to put it into one of the amoeba that typically had TACS1. All those sorts of things would be possible if we had a culture, but people around the world have been trying to culture this group of amoeba for a while and have not been successful, but we have some collaborators who are on it, and we'll see where that goes. Okay, so now I'm going to take a quick sip of water, and we are going to change gears to the bloom part of this talk. All right, so that work was at Smith College in Western Mass, and that was in a lab that was really focused on the diversity of microbial eukaryotes or protists in systems. And I came into that position and learned a ton about that diversity. Then an opportunity at Bigelow Labs opened up with this Maine eDNA project, which really just seemed like right up my alley, with looking at molecular tools to understand coastal questions and sustainability and all of those sorts of avenues of research. So I started this position in January of 2021. So I've been here a little over a year and really enjoying life in Maine and the research that's going on at Bigelow. And so the work that I'm going to present is work. So I'm working with Pete Countway at Bigelow, but this is work that's a collaboration between Pete and Karen Wilson, who's at University of Southern Maine, and her graduate student, Sharon Mann. And so Sharon won this big Maine eDNA project, which is a $20 million F-score award to the University of Maine and Bigelow Labs to really sort of take the next step in moving eDNA science forward, with especially sort of a coastal focus. And so they had a whole crop of graduate students who came into that program specifically working on eDNA questions. So Sharon Mann started maybe a year and a half ago in that program, and Pete and Karen had this existing project on a lake and asked Sharon and I to collaborate on taking a closer look at what was going on in this system. So first, I'll just talk for a second about environmental DNA. It's definitely a hot topic these days. You may have heard about this person who recently was vacuuming the air at zoos, I think, and was able to detect a surprisingly high amount of the animals in zoos by just sucking up air and collecting it on a filter and extracting DNA from that. We work in the aquatic systems, which is a little bit, I think, easier because we basically just can collect a water sample, filter that water sample, and extract the DNA from that water sample. And at that point, we can ask a lot of different questions from that water sample. So like Sharon and Karen are mostly fish biologists. So they're interested in alewives and the impact of alewives on food webs as they return to their breeding freshwater lakes and river systems. And then we're interested on the algae side. So of course, algae and microbial cells are going to get captured on that filter as well. So we can ask all sorts of questions about that. And so, yeah, that's basically what we do with environmental DNA research, but it's a field that has really taken off, but is still working on coming up with sort of standards and best practices and workflows that will allow it to become, I think, more plugged into things like, regulatory bodies and routine monitoring, all sorts of things as the cost of sequencing comes down and as technologies like, basically handheld detection of some of these methods are possible these days. Okay, so where are we working? This is Highland Lake here. It's just outside of Portland. So this is Casco Bay here. So Portland's right here, and it's not too far away from there. So a pretty developed area. It was listed as impaired, and it started blooming several years ago, and that coincided with, I believe, the restoration of a fish ladder that led to many alewife returning to that, which I would say is a good thing to have alewife returning to places where they historically have been. But if you live on that lake and you're used to a certain way that your lake looks, and all of a sudden your lake looks quite different, and it coincides with the alewife, you might start to ask questions. And so this definitely got sort of a little bit political and a little bit touchy with the folks who live on that lake. So once that bloom sort of started happening, and it would happen year after year at about the same time, folks started to really look at whether it was nutrients or food web interactions or just what was going on with the algae bloom. And so the good news is Pete was there sort of in the early days and said, hey, this seems like a good opportunity for some eDNA sampling. This was before the eDNA project even had got started. And so they went out and they collected these epilimnetic water samples, which basically means you're kind of coring down through the epilimnion, which is the top layer of a freshwater body once it's stratified in the summer. And so it might be as much as 3 or 4 or even 6 meters, and they're getting basically a core that mixes that whole epilimnion together. And then they're also collecting Secchi disk, which is a way of measuring water transparency that's been really popular tool for limnologists for a long time now. And so they, in addition, collected chlorophyll A. So this is sort of what they knew from the chlorophyll and Secchi measurements were that in late July here, so this is Secchi depth. And so if you're losing Secchi depth, that means if you go from a Secchi of 5 to a Secchi of 3, you're losing water clarity. So this is the worst point of water clarity here in late July, corresponds with this really high peak of chlorophyll A. So something seems to be blooming, something seems to be going on, but nobody knew what exactly, because people would look, they'd look at water samples and they couldn't see any like smoking gut, no really obvious cell that was to blame. And so that's where eDNA comes in handy, because we probably are working in this nano to pico size fraction, so 20 to 2 micrometers, and that's what phycologists fondly call LGBs, for little green balls, because they're just very difficult to identify with microscopy when they're that small. So we took an eDNA approach. We took a two-tiered approach that started with metabarcoding. So that's when you use sort of general primers that are designed to amplify a large diversity of life. And you do high throughput sequencing with those primers and bioinformatics, and then you get species or close to species identification, and you get these sort of relative abundance plots saying, okay, this sample had this much of this organism, this much of this organism, and so on. The other way to do these things are through qPCR, quantitative PCR. And so for that, you are designing an assay that in theory only picks up the target organism and won't pick up anything else. And you can use molecular probes and all sorts of different fancy molecular biology wizardry to make sure that that assay is really specific and sensitive. And so by combining these two approaches, we can get actual counts, whereas we only have relative abundance from metabarcoding. Here we can say there were approximately this many cells per milliliter and so on of a given organism, which is really helpful when you're trying to sort of track species abundance over time. So first we'll start with the metabarcoding, which I think I sort of went through on the previous slide, so I'm going to move on through this, but happy to talk about it more if folks have questions. So these are the results of metabarcoding from 2018, which was a major bloom year. And so the arrow here is corresponding to the sample from the lowest Secchi depth day. And so these are stacked bar plots that represent a sample and are colored by supergroup. So I also sigh at some of these names, but basically like opisthecons are animals, Archaeplastida is going to be plants, alveolata, things like dinoflagellates, and stromatophiles, things like diatoms. So this is a pretty high classification so that we can make sense of the big proportions of these organisms and how they're changing over the growing season. And what we see is that on that bloom day, you have this one big segment, which means that most of that sample is made up of this genus right here. So each break here is a different genus within one of these groups, right? So in the opisthecons here, you have 1, 2, 3, 4, 5 medium abundance things, and then a bunch of low abundance genera. But on this day, you just have pretty much one quite abundant genus of, well, in the greater plant group, but it's a green algae, and it's Rexonema, which was formerly known as Helicodictyon. Then we also looked at cyanobacteria because the bloom was not easy to identify, and so some folks were thinking that maybe it's a picocyanobacteria, which can cause water clarity issues. And so we used a barcoding approach that's going to pick up mostly cyanobacteria. And what we see are that there indeed is a good amount of picocyano, specifically synobium, but that its abundance doesn't seem to change with the date. And there doesn't seem to be quite as clear a pattern as we saw with the eukaryotic things. But we wanted to dig deeper into both of these. So that's why we then went and we said, okay, we'll design qPCR assays for the species of green algae, Rexonema, and for synobium, and really try to figure this out and get to actual counts of cells per milliliter of water. So that process was pretty straightforward for Rexonema because it's actually in the, I think the Ulvophyceae, which is a strange group for freshwater, but it is a freshwater representative of the Ulvophyceae. And so there were not a lot of other things that we needed to exclude in our analysis when designing a qPCR for Rexonema. On the other hand, Cyanobium is a really diverse genus, and so that was a little bit more difficult. This is a tree of 16S for Cyanobium from lakes across Maine, and so you can see that there are several clades in this group. So we tried to design primers that would capture maximum diversity while not including outgroup species, which is a challenge. But I think we did it. So then for qPCR, we make qPCR standards so that you basically run things of known concentration and compare how your samples line up to that standard curve. And so what we see is that for Rexonema, we see a peak in late July, and it's just over 2 million cells per mil, which is absolutely enough cells to cause a loss of water quality. And that peak lines up really nicely with chlorophyll A, and it lines up really nicely with Arcellinida. So at this point, I was like, I'm fairly confident in saying that this is a Rexonema bloom, and that's what's going on. Just the fact that that pattern so closely matches the Secchi and the chlorophyll. But we really wanted to figure it all out, so we continued with Cyanobium, and you can see that when you have the bloom day here, you don't really have very much Cyanobium in the water. And you'll see why this is scaled this way in a minute. But you can see that the phycocyanin does sort of, which is a pigment associated with cyanobacteria, does a little bit track the abundance that we're getting with qPCR. So that's the 2018 data. And then for 2019, I'm just gonna make sure I didn't miss something. Okay. For 2019, you do have a dip. It's not as pronounced as 2018, but it does seem to correspond to when you have high numbers of Rexonema in the water. And we don't have the sampling frequency for 2019 that we do for 2018, unfortunately. And you see for Cyanobium, there's not really a clear lineup with the bloom, and then you get a lot of cells sort of later in the fall, which is typical for cyanobacteria. 2021, there was not a real decrease in, so here the Secchi depth is 4, so like, yes, there's a local minima, but it's 4 as opposed to like 2 during 2018. And you do see some really clear picocyano peaks when you do have that reduced water clarity, and pretty much no Rexonema in 2021. So as soon as you start investigating a bloom, it tends to go away. I think that's how it goes in this field. And so just looking at those patterns at sort of a larger scale, you can see that we've got this broken down by year. And so 2018 in July, there was a lot of Rexonema, and by August it's waning. And 2019, there's a fair amount, but by 2021, there's really not much to speak of. And it's interesting because kind of the opposite pattern starts to happen with the Cyanobium, in that by 2021, when Rexonema has waned, you do start to see more Cyanobium coming up. So an interesting pattern there that we're interested in following up on. So just some final thoughts here. The metabarcoding data is super helpful when you are taking a first look at a cryptic system, because you don't know what you need to design qPCR assays for, any of those sorts of things, without sort of this broad metabarcoding approach to begin with. And then I would love if there are people here who know a lot about green algae. See some of them in the audience. If they've heard of Rexonema or Helicodictyon in the Northeast, it's mostly reported in sort of the mid-Atlantic states and south of that. So we are not sure whether this just went unreported, which is not super surprising given that it's a little green ball and hard to identify. But if it is causing these water clarity issues, you'd think maybe it would have come up versus could this have been introduced and not have been in Maine all along? And then once it gets introduced and certain conditions occur, it starts to bloom. I can keep hypothesizing and say, then maybe it's been in that system for a few years. And so by 2021, maybe it's really reached some sort of equilibrium. And so we aren't seeing the blooms like we did see earlier on in 2018 and before. And so then, without the qPCR, we wouldn't have really known just how many cells there were. So yes, we can say it was the majority of the sample in those metabarcoding days, but for some lake managers, unless you say like, okay, there's about 2 million cells per mil, they would say like, oh, well then, we don't think that caused the bloom because it's too small or this and that. But to be able to put a number to that and say how many cells there are is really useful. So still waiting to see if we get a Secchi depth as bad as they had in 2018. And then we also have droplet digital PCR here at Bigelow, which is basically a more sensitive qPCR technique. And so we're interested in comparing those results to just, droplet digital is fairly new on the scene. And this seems like a really good data set where we have a good grasp of what's going on with qPCR. So then we can sort of validate the ddPCR on this system. And then we're also interested in digging into the metabarcoding data to see what might be keeping rexanemia in check. So you see it sort of, pop up and then quickly decline. So is that grazing from perhaps ciliates or cryptophytes or dinoflagellates, all of those sorts of questions? We should be able to untangle by looking at sort of differences in abundance of some of those grazers in response to that bloom. And yeah, you can definitely forward your questions on to Sharon or ask me now. So I think I will stop sharing. I can always put it back up, but at least for now, I can see folks and I'd be happy to ask questions.
Awesome. Thank you so much, Robin. That's fantastic. This was great. So just a reminder, please, everybody, if you have questions, please feel free to write them in the chat and we'll read them out loud and we'll get Robin to answer them or at the very least take a stab at answering them because I imagine that this is such an interesting field where some things are still quite unknown. So I suppose while people kind of get started and start typing their questions in, I'll seed one to you right now. I was really interested in the fact that with these amoebas that you were able to starve them to the point where they started eating their own, quote, symbionts, their associate algae. And like that kind of got me really excited because that's sort of like the next step or the first step towards potentially like maybe understanding or exploring more how these organisms actually get their associates to begin with. So that's something that has always been a very interesting question in lichenology as well. So I'm seeing some parallels. So I was wondering if you could expound on that and are there any plans to explore that or has anybody done that? I know that you told us earlier that they're hard to keep alive.
Yeah, so I mean that it is a really interesting phenomenon there to observe, and it is compounded by the fact that it's difficult to keep these in culture, but I still think there's enough that you could do in the window of time that they are able to be grown. So my dream is somehow to be able to measure sort of symbiont turnover. So if you are able to keep them in a media with chlorella that are somehow marked in some way, then you could sort of watch those chlorella go into the amoeba, measure the number of days that they stay in, and then are they immediately digested? Do they ever, go into the cytoplasm and then leave again? Those sorts of questions, I think, are definitely experiments that could be done with the amount of, you can see the final day here is day 12. So I feel like that may be enough time, but would be
Do they
even
tend to
better.
live much longer than that?
Yeah. Yeah.
That's like sort of the extent.
I think so. A couple of weeks. Yeah.
Okay. Yeah. I imagine that there's, I don't know, I always hear about people coming up with clever radioisotope experiments or something like that, where they can,
Mhm.
they can like, seed an individual with some sort of detectable isotope they can check later. So maybe there's something like that. That'd be really cool.
Yeah.
We have a question in the chat. So there's a third level of genomic activity in the amoeba chlorella system, that of the chloroplast of the chlorella, as opposed to the chlorella genome itself. Is that placid activity of interest?
Yeah. Yeah. So that's a great point and an additional sort of thing that we look at. So we do see the plastid genome. So like on the starvation experiment, what we're measuring for that actually is chloroplast genome coverage. And so we are able to use the chloroplast genome as sort of a guide to tell, like, on day 0, there's plenty of chloroplast genome. We're getting 60x coverage of it. And by day 3, we're getting 2x coverage of it. So it's absolutely a useful marker of activity and something that we have incorporated. So like the RBCL tree is also working off of the chloroplast genome of that. So it is nice to have that additional piece of information. The one thing that I will say that we wish we had for this system is that the WTAs, because of the bacterial issue, we use a poly A selection for the WTAs. So we can't look at chloroplast genome transcript activity with the current data set. We would have to do this with a WTA method that does not poly A select, because chloroplast transcripts are not polyadenylated. So that was sort of we got to the end of the road and we were like, ooh, bummer, we wish we had run this with a different kit so that we could look at that in addition. But even just seeing that the nuclear genome is not there, we also have the DAPI and the autofluorescence to see, like, there do seem to be fluorescing chloroplasts in there. So yeah, hope that addresses the question.
So just to clarify, I wanted to check again because I think I might have missed that. So the nuclear genome was actually absent. You did find that it was absent. Was that accurate?
Nuclear transcripts of the Chlorella algae were absent.
Oh, right. So the activity,
Yes,
the transcription activity. Okay.
Yep.
That's right. That's right. Okay. Yeah.
So I would imagine that if you did have that ability, I would really hope that you would see chloroplast genome activity, because otherwise, like, how are those chloroplasts? I mean, another interesting question is that often, almost always, chloroplasts need a good number of nuclear encoded proteins. And so you take away the nuclear genome, that sort of, I think, immediately starts the clock ticking on a chloroplast, because its genome has some components, but some components are often still in the nuclear genome and need to get transported in.
Mm-hmm. Boy, this is such an interesting and weird relationship, because also what you observed was that so the organism, the chlorella, is sort of, it has little nuclear genome transcription activity, so it's not doing much, if anything, but it still sticks around, and appears to be alive. It's at
Yeah,
least those different organelles, everything is still visible. It's green. It
yeah,
looks like it's healthy, but like when you do starve them, you do see that like that they get degraded, they get eaten and stuff like that. So it does indicate like, yeah, this is, I don't know, like it does kind of say like, yeah, there is some sort of actual relationship here, that they're being maintained. Like maybe they're just being maintained as an emergency food source, but
yeah.
that's such an interesting situation. That's very cool. Um, so we'll leave this open for a little bit longer for people to come and ask more questions. But um, I was also hoping that you could explain to everybody what the process of selecting an individual cell for single cell sequencing is like.
Well, is it okay to talk about mouth pipetting in 2022? Do we still talk about that, or do we just do it behind closed doors?
uh,
So that's
well,
the key is mouth pipetting.
okay.
So, and I didn't do that work. That was mostly Agnes, and then some of the undergrads are amazing at that. And it's just, time under the microscope, and really, you're heating up your glass pipettes and stretching them out so that you're getting a really, really fine point on that pipette, and then you're just able to grab one of those cells, wash it a few times, and put it in the tube. So
Yeah.
like I said, like that fine scale work is not necessarily my forte, so I really leaned on those folks to do the actual picking of those cells.
We have Fabian's seal of approval here behind closed doors.
They actually, for the so the NCMA is here at Bigelow, and they host a lot of courses on algal culturing and identification, and they have a really neat system where they basically have like it's a bulb and then a like a screw, and so it's basically just recreating a mouth pipette, but instead of your mouth, it's like the suction of this bulb and a little screw, and so you can like put that in and release that and then suck something up. So it doesn't work quite as well, but at least for sort of a class when you're showing people things, maybe it makes them a little bit happier than mouth pipetting.
Well, that's still really cool. Let me see. Another question from Fabian. How prevalent are chlorella in those bogs? In other words, is it easy for the amoeba to find them?
Yeah, we haven't explicitly tested that, but I would think that they're pretty darn abundant just because of their size, and chlorella tend to be this group that's just somewhat ubiquitous. I will say a couple interesting things that brings up. Number one is that in the read mapping data, we were able to see that you pretty much never had a mix of the chlorella types in an amoeba, right? So you can imagine like, okay, you have TACS1 and TACS2. And from our biogeography work, those occur, one occurs at Harvard Forest, and we'll call that, that's TACS1 at Harvard Forest, and TACS2 is at Holly Bog. Those bogs are 20 miles apart, maybe less. And so the fact that there isn't the chlorella at the corresponding bog is a real head scratcher. And that even if it was there, you never seem to see an amoeba that's taken up some of each of them, even though these bogs are so close to each other. So there's really some interesting both biogeographic patterns of the algae, which I think we should do, and we could do immediately. We could basically just go collect sphagnum, not the actual amoeba, and ask, meta barcode that, or just do a metagenome of that and say, okay, how many chlorella genotypes do we have at Harvard Forest, and how many chlorella genotypes do we have at Holly Bog? Then if you do see a really diverse assemblage, then the big head scratcher is, well, why are the amoeba always seem to be taking up just one type? And so that sort of preferential feeding, I think, opens up a ton of interesting questions. Which is why it would be great to have a culture so that then you could actually test that and try to feed them the wrong kind.
Yeah. Yeah. That's really cool. And certainly like in other groups that have this sort of association, like in lichens, we do see like some degree of like partner selection for specific genotypes that you know are better in like certain environments and stuff like that. There's a cool study in Japan that did something very similar, just like kind of exploring that. So maybe there's something like that going on. That'd be really cool. I have another question. I found paramecium with presumably Chlorella endosymbionts in a local marsh, northwest Pennsylvania. Is it known whether that relationship is mutualistic on the way to a true permanent endosymbionts, or kleptoplastic, or unknown?
Yeah, so I think the paramecium actually shows up on this figure from I think it's a Decker paper from 2009. And so they classify it as sort of a persistent symbiont. So it's not obligate because you can find paramecium without it, I believe. But it is what they term as like persistent. It's almost always there. There may be a life stage of the paramecium where they don't have it, but given time, I think the paramecium almost always, that group of paramecium almost always acquire and keep those. So yeah, it's not kleptoplasty. It's definitely more stable than that. And these are the fascinating things that you're like, okay, give it, like, let's run the simulation, give it 1 billion years to those just become, is paramecium then a photosynthetic organism? You know, probably. Why would you, once you've figured out that hack, why would you ever give it up? If you could photosynthesize, wouldn't you choose to?
Right. I would. Where's my chloroplasts? Chris would too. Yes, we're in agreement. I'm going to ask a speculative question because I'm curious what your thoughts are. So you and I were talking briefly about this before we started today. That Chlorella, or you know, green algae generally, but you know, also Chlorella specifically, has a lot of different associations beyond this one. There are some lichen associations. There's obviously this one. There's the paramecium association. I think this is also what is associated with certain radiolaria. I was curious if you had some thoughts on why Chlorella or like green algae seem to keep such diverse associations. I don't know if I said lichens, but lichens too are in there.
Yeah. I feel like it has to at least be related or involve transport across membranes, right? So if you can figure out, okay, so number one, if you are a heterotroph that acquires one of these, you would need to make sure that somehow nutrients are coming out of that system. But for some of these more stable systems, you probably need to be getting signals perhaps into that system, or certain, I don't know, molecules that are going to make that algae that you're housing happy. So I do feel like something about transport systems in Chlorella across membranes must have to play some sort of role. But yeah, it's a really great question that I don't know if I have a satisfactory answer to just why they're the perfect symbiont for everybody.
Well, I mean, I think your answer was pretty interesting, so I do like it. obviously none of that stuff is known. So it's all speculation at this point. Super cool. I've got another question, maybe off topic, but I like, I like that science mention.
Now.
Just for the it's just for the audio, just for the video later. I'll read this out loud. Maybe off topic, but I like that sigh at the end of at the mention of the current eukaryotic super kingdom designations. Do you think this taxonomy will be relatively stable, or will we all have to learn new names in 10 or 20 years?
B.
B.
I'm gonna go with B. No, I don't. Sorry.
I'll prepare myself.
Yeah, I think, I mean, in some ways that's going to be cool, because maybe it means that we realize something that we haven't realized, you know, in terms of the relationships between these supergroups. But in a lot of ways, I think it's moving the deck chairs around on the Titanic sometimes.
Awesome. Sorry, I had to turn my video for just a second, but awesome. Thank you so much. Thank you so much, Robin, for giving us all your time today, answering all these questions. I found it super interesting. I hope everybody else likewise found it super interesting. This is such an interesting conversation, and I'm going to research this on my own a little bit later to learn more about these relationships. So again, thank you, and thank everybody, all of you, for coming and being a part of this for our May Torrey Talk. We really appreciate having you all here.